Group of people I want to ask about here is, are there anybody in the room that have had cancer or survivors of cancer? We got one. And you don't have to raise your hand if you're shy, but that's another reason I want to do this, is to bring some awareness as to why scientists are working hard on a day-to-day basis to help cure cancer. And that I'm going to get into a little bit into why I got into science as well. But first, I want to introduce you to someone else. This is the antibody. This is one of the most exquisitely designed molecules that nature has ever come up with. It's a quite striking molecule. From the tip of the fingers of the arms down to the toes, you've got 440 amino acids connected like beads on a string that make up the heavy chains. There's a yellow one there, and there's a teal or blue teal colored one. Those are called the heavy chains. And then you have some light chains, which are the smaller ones that are about 220 amino acids. And they're up in the arms only. There's a purple one, and there's a green one. This protein is made up of 4 polypeptides that create what's called an antibody. And they have exquisite properties that make them amenable to being used to help treat cancer. You know, growing up, people would ask you, what are you going to do when you grow up, right? And I went through the phase of wanting to be an astronaut and wanting to be a policeman, a SWAT team member, and so on. But in high school, when people started asking me that question, I started to say, I want to find a cure for cancer. And I started saying that. I'm not exactly sure why, except that I wanted to do something big. There was no one in my family that had cancer. I wasn't impacted by it, but I knew I loved science, and I knew I wanted to do something really big. So in high school, I started to say, I want to find a cure for cancer. People would look at me like, what's the matter with this kid? You know, he had better luck being an astronaut than finding a cure for cancer, right? So but I kept saying it, you know, and I kept saying it while I was at the University of Waterloo in Canada, getting my bachelor's degree in biology and chemistry. I said it for 4 years throughout my undergrad career. Then I came to USC. I went into molecular biology and getting my Ph.D. in that field. I wasn't working on cancer, but I was getting some skills in place, and I still wanted to find a cure for cancer. I was doing genetic mapping studies, and then I moved to Cedars-Sinai fellowship with UCLA and did a postdoc, and I started doing Down syndrome research and doing physical maps of chromosome 21 and trying to identify genes that were involved in Down syndrome. Still not doing any cancer work, but when people ask me what I'm going to do when I grow up, I kept saying, I want to find a cure for cancer. So for about 15 years, I was saying that, and I still wasn't doing that kind of work. And it wasn't until in 1997, after I left my postdoc, that I helped start a small company in Santa Monica called Agensys. And I became a gene hunter because I was hunting genes for Down syndrome. But now I was finally hunting for genes that might be involved in cancer. So my mantra finally came true. And I didn't know at the time when I was in high school what a mantra was, but I realized looking back that that's what it was. I kept saying it over and over again. And I think that's a lesson for the young children and the old in the room, that you got to verbalize what you want to do in life. Find a simple phrase that you want to do. You want to find a cure for cancer. You want to find a cure for Alzheimer's. You want to travel to the moon, you want to travel to Mars. Just find that one saying and keep saying it over and over and over again. It took me 15 years before I finally started doing that work, but if you have that passion and you keep verbalizing it, it will come true. I think that I'm an example of that. So that worked out real nicely for me. So I worked at Agensys. It was a startup from UCLA. We had 25 scientists at the time. And I became an inventor on over 110 patents that were all about genes, hunting genes that were involved in cancer. And then I moved to Xencor. In fact, I've worked at Xencor and Agensys twice. I went back and forth between those two companies. And about a month and a half ago, I got laid off from Agensys. But yesterday, I signed an offer to work at Amgen, and I'll be starting there on October 9th. So I got through the perils of biotech, which could be unstable, but now I'll be continuing my career at Amgen. Okay, so let's move on here. I want to keep this fairly informal, and I don't want to blow your mind with too much science. So I got a few videos in here that kind of describe the life of scientists. I've got some science. I've got a few things that should be interesting for you to hear. But please pipe in if you have any questions. This video here is about 1.5 minutes, and it's a pretty special video for me, and I'll talk to you about it afterwards. But I'm hoping you'll listen to the words carefully. It's a poem by Dylan Thomas that you may have already heard, but it's got a great message that impacted me a lot, and I want to get your opinion after you listen to it. So let's see what you think about this.
Do not go gentle into that good night. Old age should burn and rave at close of day. Rage, rage against the dying of the light. Though wise men at their end know dark is right, because their words had forked no lightning, they do not go gentle into that good night.
Seen this commercial before? If you have, raise your hand. So we got two or three people, and 2 of them are scientists. Now I'm sure everyone in this room has seen this video. This commercial. It was on TV for over a year. But like a lot of people, and my wife's the same way, tunes out a lot of commercials when you're watching them. You know that's what TiVo is for. But for scientists in particular, it affects us a lot. To keep it simple, just try to remember 3 things today here. So the first thing, I'm going to talk a little bit about cancer, give you a little bit of things to remember what cancer is, but specifically, what are antibody therapeutics, which is basically my background for over 20 years. And also want you to understand that scientists are fighting a fight for cancer, against cancer, and they're in your neighborhood, actually. You may have neighbors like Misty, who's a cancer fighter, and there's other people in the room that do this. I'm pointing out 3 companies that I know a little bit about. There we are at Topanga Library in red. But Amgen is up in Thousand Oaks. It's a big company, about 5,500 people, and they don't only do cancer research. They fight other diseases as well. Xencor is at the other end of the LA County there. They're involved in engineering antibodies for cancer. And then Agensys is in Santa Monica. Again, a company that's totally focused to fighting cancer with antibodies. This is their campus here in Santa Monica. It's ability to develop monoclonal antibodies and monoclonal antibody-drug conjugates, which are called ADCs, to treat cancer and to provide therapeutics for unmet medical needs. Amgen strives to serve patients by transforming the promise of science and biotechnology into therapies that have the power to resolve health and save lives. In everything we do, we aim to fulfill our mission to serve patients. So it's a very noble profession that we're in, and we're really doing this to help patients. And Xencor has a simple mission of redesigning the antibody from the bottom up in order to make it a better therapeutic. To get to know scientists a little bit better, I have a series of 3 videos point out that scientists have lives outside of science.
As a kid, I always saw horses in the pasture. I would tell my mom, "Look, horse, horse." I want to stop and see the horse, pet the horse. And I get that passion from my mom, I believe, because she was also in the horse business as a young adult. At 9 years old, she finally gave in and got me riding lessons, and I loved it. I fell in love with riding horses and just being around them. At the end of a tough week, it's nice to go up and pet a horse. It's very relaxing. When I'm riding, it's a combination of feeling relaxed, but at the same time, you're controlling this 1,000-pound animal. So you have to be alert. Horses can't talk to you, but they do communicate. They give you cues about how they're feeling when you're on them. When you become calm and relaxed, the horse will follow your lead and also become calm and relaxed. The world seems better, definitely, on the back of a horse. Trail riding, I think, is the most relaxing because you're out in nature. You can hear the birds singing. You get to go exploring areas that you may have never been able to explore before. You feel more connected when you're with nature. There are things I do with a horse that are applicable to what I do in the lab. With a horse, there's a lot of trust and teamwork that's involved. In the lab, teamwork is very important because I may know how to do one aspect of research and my colleague may know how to do another aspect. And it's the collaboration between the two to get all the information we need to get the job done. I work in protein technologies, and our specific area is making antibodies to different cancers. And my specific job is to produce the antibodies in my group. Antibodies are the body's natural immune response to anything foreign that comes into the body. And we can engineer these antibodies to target specific cancers. I have a history of an autoimmune disease, and so as a child growing up, I was in and out of the hospital. When I go into work, almost every day I'm reminded of the medicine I took when I was a child that helped make me better. Having that personal experience helps keep me focused on what I'm doing and the people that we're helping by making these types of medicines. These therapeutics that we're making can be tailored to specific cancers, specific patients. They're tailored medicines, you know, it's not just chemo to cure all cancers, it's specific therapies to cure specific cancers. I'm very satisfied with the career path I've chosen. I believe that cancer research is paving the way for bigger and better therapeutics. I enjoy it because I'm helping people this way. I'm not a doctor that's helping individual patients. I don't necessarily get to see the end product, but when our medicines do go out there, you know, it just takes one to treat many different people.
Just a reminder what cancer is. A simple definition of it is a biological consequence of unregulated cell growth. So you usually get a mass. Basically the cells, these are cells gone wild. They just kind of take over. They grow where they're not supposed to grow. They're part of you. Cancer is your own cells that go awry. It's not like a foreign bacteria that's growing in your body and causing problems. It's your own cells that wind up having problems, and it can cause ulcerations, fractures, neurological symptoms, fatigue, all sorts of things, and ultimately death. So cancer is pretty nasty. One way to think about cancer that I think is useful to remember is you can compare cancer cells to cars. This monster car here in red that's kind of crazy looking. You got the ugly green cancer cells on the left. So a cancer cell has constant proliferative influences. That's kind of like having the gas stuck on your car. It just keeps going and going and going. And to make it worse, your brakes don't work on your monster car. So the cells don't have any suppression activity. So they just keep growing and growing and growing. And this cancer cells also avoid death. And then that's as if the monster car can avoid crashes by driving around them and so on and being evasive. And cancer cells are immortal. It's like as if the monster car would keep going and going after multiple collisions. And the monster car can go off-road, can drive anywhere it wants, and that's what happens with cancer cells. They just kind of go all over your body in places where they don't belong. And the cancer cells also evade your immune system. And that's as if the monster car had a radar detector so the police can't catch up to him. So, and the cancer cells have a constant blood supply. They develop their own neovasculature, it's called, so that they can have constant blood supply. And that's as if your car never runs out of gas. Kind of a comparison between a monster car and a cancer cell. Help you remember what key aspects of cancer. In terms of causes of cancer, what we put in our body has a big influence. Tobacco is a big source of cancer, as well as diet and obesity. 2/3 of the risks come from things that we can control ourselves. Infections, viruses, and so on, hard time controlling those, and there's other factors that come into play. But a healthy lifestyle goes a long way to helping to reduce the risks of cancer. Here's some examples under the microscope of what cancer would look like. So on the left, you have normal example of bladder and colon tissue. The purple and dark purple colors represent nuclei of the cells. So you can see on a normal bladder that there's a row on top of cells that are neatly organized like soldiers lining up all along the top of that. That's the transitional epithelial layer. When you have cancer, those soldiers go all awry. That whole field is just full of cancer cells that are not organized anymore, and that's a hallmark of cancer. You lose organization. You see that at the bottom as well, normal colon tissue compared to the cancer tissue. The villi structure of the normal colon get all disorganized. In breast cancer, you see the same sort of thing as well. There's different types of cancers here, ductal carcinoma on the right. And as the cancer gets more and more aggressive, you see more and more purple colors, meaning more and more cancer cells growing and dividing in a disorganized fashion. Here's prostate cancer, where the prostate glands start off grade 1 position as being very rounded and organized. And as you go down to the grade 5, they get uglier and uglier. And you can see that in the cartoon on the right as well, that as the grade of cancer increases, the glands get more and more disorganized and de-differentiated is what the word is for that. So that's a look at what cancer cells look like. Here's a second of our third scientists. Now, this Pat is from Zoma, which has a history in the LA area as well. Zoma started in the 80s at the same time as Amgen. They started in Santa Monica, actually. The funny thing is when Agensys started, we used their facilities, the same ugly red brick building. They used to make airplane parts in the 50s, got converted into a lab, and we shared their lab space at some point. Now Zoma is moved completely to Berkeley. They're no longer in the area.
See, I got my Ph.D. in chemistry, and I started blowing stuff up when I was 8 years old. I mean, you know, I got a chemistry set, I think, in 2nd or 3rd grade. I love what I'm doing. There's never been a boring day. Some days are good, some days are bad, but I can tell you, it is exciting. We're looking for novel therapeutics that change the course of disease that have no alternatives. What we're interested in is not just killing cancer cells, because I can pour acid on a cancer cell and kill it. The idea is to kill the cancer cell without killing the other tissues so that the person ultimately is able to get rid of the cancer and live a healthy life afterwards. Seeing how the scientists individually work and work together as a team toward that goal is very, very exciting. I get goosebumps. There is a great deal of satisfaction and joy in taking your knowledge and using it to help other people. There is no doubt there is a connection between what I do in my work and what I do in the time away from work. There's a small team of us called the Bent Prop Project, as in bent propeller, because that's the only kind of propeller we ever see. Then on an annual basis, we mount a mission. We go out into the South Pacific and look for remains of Americans who've been missing in action since World War II. For 20 years, I've made that a very important part of my life. It involves actually a lot of what I do in science, only very different application. We also have a group of high school students that are joining us. They're part of an advanced robotics team. It's like being a detective underwater for a purpose, and you're looking for the smallest clue that might be the beginning of a debris field that leads to an aircraft. All of that has to be planned out just the way you plan out an experiment. It's not just about finding the remains, it's getting them home. Ultimately, it connects back to bringing back information to family members, which is not too dissimilar from delivering therapies to patients and seeing benefits that they couldn't get otherwise. It's not just about doing science, but science with a purpose. As Chief Scientific Officer, I'm always excited about progress, and any little step forward is a big day for us. Zoma's in the area of biomedical research. We develop new therapies to help people.
Antibodies have been around for about 125 years. This is a timeline here, starting back in the 1890s, where Kisasanto and von Behring were one of the first to talk about this concept of antitoxins and passive immunity. And Ehrlich coined the term magic bullet, which we've all heard, right? The magic bullet travels through your body, it only kills the bad guys. And he coined that term. And also the term antibodies, which we're using today. He was a German fellow, and all three of these have won the Nobel Prize along the way. And Ehrlich helped develop the diphtheria antitoxin, which I'll talk about in a little while. So Heidelberg was the first to actually say that these antibodies are proteins. So before that, it was just a mythical thing that was happening. They didn't have it identified as a protein. So Heidelberg described these as proteins. Later, in the mid-1900s, Pauling confirmed this lock and key theory that I'll talk about, and the discovery that antibodies are made by B cells came out during this time frame as well. And then the structure that you saw that I was rotating came about in the late 60s, and also more about the origins of the antibody diversity, which is an important factor of antibodies. And Calder-Milstein got Nobel Prize for this hybridoma technology that actually my lab used quite a bit to develop antibodies. That's how antibodies are generated in the lab. And then the very first drug that used an antibody was in the late 1980s. It's a protein on T cells, and it was used in graft versus host disease. Whenever you had organ transplants, people would lose their organs. And that was the very first antibody drug that was ever used. And then in the late 1990s, the first oncology antibodies came about. Rituxan and Herceptin, both two very effective antibodies that are used today quite effectively. Herceptin's for breast cancer, and Rituxan is for lymphoma and leukemias. So since that time point, this is a little outdated here, but there's probably over 400 antibodies that are in the clinic at this point. So it's come a long way in 125 years, and it's helped save a lot of lives. Here's an interesting picture of a vial, actual vial of a diphtheria antitoxin. Well, diphtheria was killing children and young folks by the thousands, tens of thousands, at the turn of the century. And it's a bacterial infection that released a toxin that would affect respiration, heart, and organs, and would be very nasty to children. And Ehrlich was the first to actually develop a commercial process for making this antitoxin. So the antitoxin at the time, and actually still does, came from injecting the antitoxin into a horse and having the horse develop an immune response against the antitoxin. And they would just simply collect the blood. That's what they're doing there, collecting the blood from the horse. The horse was fine. They would spin down the blood, collect the serum, and actually inject the serum. That's what you would take this vial, put it in a syringe, and you would inject that into a patient who had been infected with the diphtheria. And it saved lives, and it was quite a huge accomplishment. Animal, and it's also very large, so it had a big blood volume. So they were able to, with a single immunization of a horse, the horse was very resilient to the toxin, would survive fine, but its body would make antibodies against it. So they would collect a lot of blood and make a lot of vials like this that they would be able to sell and distribute to the hospitals. This process is still used today. There's a commercial antitoxins that you can purchase today that come from horses. In fact, botulism, black widow spider bites, and these are still antitoxins that are generated by getting serum from immunized horses, and sometimes it's humans that are used as volunteers. So what's in that anti-serum? Well, it's antibodies. That's the trick. It's a whole bunch of antibodies that are binding and neutralizing the toxin. That's what you find in the serum. It's a polyclonal, we call it polyclonal population. It's a mixture of all sorts of different antibodies fighting the toxin that might be present in your body. I talked about this lock and key concept earlier. So remember, the antibody is made up of 4 chains. There's 2 heavy chains and 2 light chains. And each antibody is very, very specific for a particular protein. One antibody will bind one in multiple billions of proteins, and it's very, very specific. In this case, the yellow antigen is the one that binds this antibody. And it kind of forms this lock and key concept that helps you remember how an antibody works. Only one key opens one lock, only one antigen binds one antibody. And on the left here, I just show you more structural formats here, that there are multiple peptides on the top left, but only one will bind to the antibody at the locations that I've indicated there. So the targets for antibodies could be a lot of things. It can be viruses, bacteria, proteins and protein fragments, cell surface proteins for therapeutics. And that's where in cancer this comes into play, right? If you have a protein that's overexpressed or only present on cancer cells, but not on normal cells, then you have an ideal target for an antibody. Sugar complexes could have antibodies against them, DNA and RNA, any man-made compounds as well. So all sorts of things could elicit an immune response for antibodies. And we actually, in our group, made antibodies against small molecule compounds as well. Even lead, there's a company, like little kits for measuring lead in your water. They had antibodies against lead, and that's how their little kit works. So here's a little cartoon of a good target for a cancer antibody, right? Here's a picture of a cell, and you can see in purple there's an antibody. And you can see that when the antibody binds the protein on the cell, a process of endocytosis brings in the antibody into the cell. It's kind of like a Trojan horse thing that's going on here. So now your antibody is not floating around in space, it's actually inside the cell. So it can create some damage, hopefully, to the cancer cell at that point and kill the cancer cells. Brings me into the next part of my talk here is that naked antibodies, we call them, just antibodies by themselves, may not necessarily be potent enough for some cancers. And a way to make antibodies more potent is by attaching small molecule compounds that are toxic to cancer cells. And that's what's depicted here with those yellow spikes. Through chemistry, you can actually attach chemical compounds to antibodies. So the antibody finds the cancer cells, gets inside, and then releases the toxin and then kills the cells.
In recent years, a new generation of engineered antibodies have begun showing promise therapeutically, particularly for cancer. Antibody-drug conjugates exploit an antibody's specificity to deliver a cell-killing agent directly to the target cells. This potent package is made of 3 essential elements: the antibody, or antibody fragment, a linker, which can be cleavable or non-cleavable, and a cell-killing drug, or warhead. Depending on the choice of linker, there are dozens of cysteine and lysine residues available to link the antibody to the drug. So each antibody-drug conjugate requires careful planning. The amount of drug bound to each antibody must be just right. Too much and the antibody may not function. Too little and the effect may not be potent enough to kill the target cells. Take the breast cancer drug trastuzumab emtansine, also known as Kadcyla, as an example. It's made up of the monoclonal antibody that forms trastuzumab, also known as Herceptin, joined to the chemotherapy drug DM1 via a non-cleavable linker. In HER2-positive breast cancer, the antibody binds to HER2 molecules that are overexpressed on the tumour cell's surface. Once bound, the lethal package is internalized through a process called endocytosis. Inside the cell, the antibody-drug conjugate is delivered to the lysosome and broken down. This releases the drug into the cell, where it's free to disrupt the cell's mitotic machinery, triggering apoptosis. The cytotoxic drug can also diffuse into neighbouring cancer cells to afford more cell death. a process known as the bystander effect. There are over 100 clinical trials underway for various cancers that are testing different combinations of antibodies, linkers, and warheads. And with research focused on new technology to develop better linkers and targeting, the future of antibody-drug conjugates is looking brighter than ever before.
Yeah, so this is, again, as I mentioned, is an approach to making a naked antibody more toxic. These are the things that are coming along and being perfected all the time. So as I mentioned, you got the killing power of a chemotherapy agent with the targeting advantage of an antibody that creates a killing machine that hopefully will be more specific to treating cancer. Some of these killing agents come from nature, actually. This dolastatin compound on the top right came from a sea slug, actually. It's a very toxic compound, so you don't want to step on the sea slug by accident because it'll kill you. The toxin was actually purified and actually attempted to be used in chemotherapy regimens, but it was actually too toxic and it would, the patient wouldn't do well on it. But when it's attached to an antibody, as it is for Xencor, it actually works well and it is used to treat cancer. Taxall is another compound that comes from the Pacific yew tree. So it's another natural compound that can be used to treat cancer. Taxall is not used in ADCs, but I just want to give you an example of some toxic compounds from nature that are used to treat cancer. This is a rendering of the auristatin compound that's derived from dolastatin from the sea slug. You can kind of see the evolution of antibody medicines, right? You got, at the turn of 1900s, you've got this anti-serum from horses. As the hybridoma technology evolved, we're able to isolate monoclonal antibodies that only bind one protein, and then recombinant DNA technology, and then also now to the antibody-drug conjugates to make the antibodies more toxic. Another way of doing it is to start off chemically and doing it chemically. Like, you know, aspirin comes from a tree bark, and the Native Americans used to chew on the tree bark when they had a headache to get the aspirin out of it. Luckily, we were able to synthesize aspirin from scratch in a chemistry lab. You don't have to chop down a whole bunch of trees to get the bark from it. So here's a list of approved therapeutics that use antibodies. So the first category is oncology. So there's 26 antibodies on the market. These are the naked antibodies that are used in all sorts of different kinds of cancers. There's a lot of non-oncology indications, actually, for autoimmune disease. Actually, Humira, which you see commercials of all the time on TV, right? That's an antibody that's not used for cancers. It's used for arthritis and Crohn's disease and so on. That's one of the number one or number two selling pharmaceutical in the world, actually. If I think about the 4 or 5 of the top 10 pharmaceuticals that are being sold are actually antibodies. So you may not known about them, but they are an emerging modality for treating disease, and they keep getting stronger and more prominent. There's only 2 ADCs on the market right now, one for breast cancer and one for Hodgkin's lymphoma. This is a little outdated, but there's probably over 400 antibodies that are in clinical development as well. So there's a lot of them coming down the pike that are in phase 1, 2, or 3 clinical trials. And there's over 100 antibody-drug conjugates that are in clinical trials as well. So there's a lot of work being done. Here's a list of some marketed antibodies for cancer. So Erbitux and Herceptin are used for colon cancer and breast cancer, respectively. They're often used in combination with chemo. Kadcyla and Xencor are the 2 ADCs that are on the market as well for breast cancer and advanced Hodgkin's lymphoma. With Agensys, these are the programs that are in the clinic, actually. Some of them are inactive, but you can see that some of these ADCs, a lot of them are ADCs. So the company was heavily focused on ADCs. Actually, Veronica, in the back of the room there, was involved in that group at Agensys. And so we got all sorts of indications from pancreatic cancer to bladder cancer, lymphomas, and so on. So if you want more information on these, you can see them at clinicaltrials.gov. One thing to note is the ones with the asterisks here, these guys over here with the asterisks, I'm proud of because I told you I'm an inventor on over 110 patents from Agensys. These were genes that I helped identify back in 1997 to 2001 that eventually have gone all the way to the clinic to be targets for antibodies. So it's really satisfying for me. And there's one that's not on here as well, that Genentech licensed from us, that is an ADC that's also going through clinical trials. So it's very satisfying to see things make it to the clinic and hopefully to market. It takes a long time. Like I said, it was back in 1997 they started this work. So that's why it's a long, arduous battle that we're fighting here against cancer. Summarizing the 3 companies that are in the greater Los Angeles area and how many antibodies they have in oncology clinical trials. So Agensys has 5, Xencor has 2, and then plus 4 that they've partnered with other companies, and Amgen has 8 antibodies that are in the clinic right now. So there's 19 just in the LA area that are going through clinical trials. So that's pretty impressive. You know, people think that LA doesn't have a biotech hotbed like San Diego or the Bay Area, but we have a lot of good science going on in the area. It can take a long time. You go through various stages of identifying a target, validating it, making the antibody, doing the chemistry for the ADCs, and doing the testing and getting it into clinical development. In clinical trials, you start off with an IND, investigational new drug filing with the FDA, and then you can go to a phase 1 clinical trial, which takes about 1.5 to 2 years with 30 to 60 patients, and then you go on to phase 2 if things look safe, and you increase the number of patients. It could take 10 plus years to get something to market. It actually gives you a hint about what's coming, what's the future of antibodies, and what's the future of fighting cancer.
This is not a video game. This is not a screensaver. This is the destruction of a cancer cell by the body's own immune system, thanks to medicine that didn't exist until now, and today can save your life.
So here at the beginning you saw a T cell killing a cancer cell. Now the Kite technology could be applied to that little video. I'm not sure if that's what they were using there, but also antibodies can be used to do that. So there's a concept that was called a bispecific antibody, and that technology is developing right now, where one of the arms of the antibody is binding the T cells and the other arm is binding the cancer cells. It's basically bringing the cancer and the T cells together in a close proximity so that the T cell can kill the cancer cells. So that's improving your immune system to fight cancer. And that's something that's in development. Amgen is involved in that technology as well. That's my second bullet point there, helping the immune system to kill cancer cells. That's the future of antibody therapeutics as well. So these bispecific antibodies engage the T cells and bring them in close proximity to cancer cells to kill the tumor cells. There's also antibodies that will activate the T cells, hyperactivate the T cells so they're more potent. And there's also this concept of using antibodies to prevent inhibition of T cells. These are checkpoint inhibitors. There's national mechanisms for making sure that T cells are not too potent, and that cancer cells use that mechanism, unfortunately for us. So these antibodies overcome that mechanism. So there's a lot of great things coming on. The antibody-drug conjugates are getting better and safer, and more of them are in clinical trials. So there's a lot of good things coming down the pike. So I want this to be kind of an optimistic ending to the presentation, that a lot of progress has been made in treating cancer here, like the first bullet point here. So the death rate has fallen 23% from its peak in 1991. A lot of that has to do with new medicines coming on the market, including antibodies. And the survival rate for childhood cancers has increased quite a bit as well, which is great. There's also this personalized medicine emphasis that's happening right now as well. And there's 23 million years of collective lives being saved since 1988 to 2000. So there's been a lot of progress. You may hear a lot about the tragedies and the downside of what's happening with cancer, but we've come a long way. We still have a long way to go, and we're still fighting this fight. In this chart over here, you can see that if for breast cancer, if you detect it when it's localized, you have a 99% chance of surviving 5 years. If it starts to spread a little bit, that drops down into the 80s, and if it's already metastasized, your chances are very slim of surviving 5 years. So early diagnosis is really important for cancer, breast cancer, and this is in colon cancer as well. The same sort of thing. You're up into the 90s. If you detect it when it's localized, you can survive 99% chance of surviving 5 years. And as it spreads, your chances of surviving 5 years drop. And then that applies to so many other cancers. Melanoma, same sort of thing. And prostate cancer, look at that, prostate cancer. Even if it's spread a little bit, you have a 100% chance of surviving 5 years. But if it's gone to distant sites, you have a very slim chance of surviving in the next 5 years. So early diagnosis for prostate cancer, getting a prostate checked, you've got breast exams, you've got colonoscopies, you have all these tools out there that could really be a big benefit for you, as well as healthy lifestyles and so on. So make use of early diagnosis is a good thing. The cancer rates have been improving for quite some time here, and they continue to improve, the cancer survival rates. Unfortunately, lung cancer, it's a very tough cancer, and it hasn't changed very much over the years. I think this is my last video. This is the third scientist in the series, and Dowdy also worked at Agensys with Misty and I, and here's his story.
My passion from flying really comes with this whole theme of adventure, this whole theme of trying to look at something differently and do something differently. I remember as a kid looking up all the time, looking at the planes flying overhead, just trying to figure where are they going today? Maybe they're going to South Africa, maybe they're going to South America. It's an adrenaline rush for me. I like the whole fact that it's freedom up there. You get to see things from a completely different perspective when you're in the air. And I think science allows you to do that as well. Science allows you to see things from a different perspective. One of the things I think as a scientist is that we are explorers. We try to understand how biology works, and especially in our case, to understand how biology of cancer works so we can develop better therapeutics to treat people. The primary goal of Agensys is to develop antibodies or antibody-drug conjugates, also known as biologics, to treat cancer patients. My job there is I lead the antibody-drug conjugate group. The focus of our group is primarily to really understand how these antibody-drug conjugates work in a preclinical setting. And we also try to determine how we can use these drugs in a preclinical setting that would help drive the clinical outcome of these drugs. What that means is that before these drugs actually can enter into the patient, into the clinical setting, we need to have a very good understanding of how they work in our model systems. Really what we want to do is to try to develop the best therapeutics that we possibly can develop. My grandfather passed away with lung cancer. I was a little kid, maybe 8. I didn't really know what cancer was. I just know it wasn't good, and they killed my grandfather. So, you know, I've always been interested in doing cancer research because I really wanted to make an impact on someone's life. So for me, that's what keeps me going, things like that, where you can see that some of the things you're doing in lab have a direct impact on how someone lives their lives. And particularly in the case of cancer patients, you certainly want to be able to impact their lives such that this person can live longer and hopefully with the drugs that we develop, live even healthier. I'm an African-American scientist. There's very few of us around, especially as pilots as well. And I want to make sure kids that are like me see someone who's like me. I'm here, I'm like they are, and you can do the same thing too.
All right, so let's recap a little bit what you need to remember, right? So what is cancer, right? It's this ugly supercar that's doing a lot of crazy things. It's unregulated cell growth. And you all can recognize an antibody now. If you see anybody walking down the street, you'll know right away, right? And then you heard about antibody-drug conjugates. And you heard a little bit about local biotech, including Agensys, Amgen, and Xencor. And I hope you have an appreciation for what scientists do behind the scenes. We're not celebrities. You know, we're not sports figures or actors and that sort of thing, and you really don't hear about scientists making big contributions to science. So, but we do, though, and we really are fighting on your behalf for a cure for cancer. And then the last thing, I mentioned my mantra here. I hope you can think about your mantra going forward as well, and and be conscious of that. That I think it can really change your lives, and it did for me. So I want to thank Topanga Library and Misty and Oleg for for working this out for me. And Misty set everything up. I just showed up here, and then also to thank the biotech scientists in the room also. And I want to thank my wife, Leanne, who's here today. She's been with me for 28 years, and she's heard me say that mantra over and over again. Actually, we met the week before I started my Ph.D. at USC, and she's had to listen to that mantra over the years. She's had to she shared my successes and my challenges, and the recent challenge of being laid off, and but luckily overcoming that, and I'm starting my job at Amgen on October 9th. So I'm pretty excited about that. So the future looks bright for me, and it looks bright for taking care of cancer.